to increase the likelihood of their research being reproducible. After this point, I do want to jump into two very specific areas in which reproducibility of studies can oftentimes be improved. First, we will focus on microenvironments that cells face and how to ensure that cells are not stressed day in, day out through normal lab actions. In the last topic, we will look at how machine and software automation can remove the vagueness of the human element and how this can lead to better and more reproducible research. Just as a side note, as we go through these last two sections, we will spend some time looking at the options that Synteca itself offers to remedy some of these issues we mentioned. So with that, let's jump in. Okay, so what do we know about reproducibility? Well, first and foremost, a simple definition of reproducibility would be something like reproducibility refers to the capability of a researcher to duplicate the results of a prior study using the same materials as were used by the original investigator. That is, a second researcher might use the same raw data to build the same analysis files and implement the same statistical analysis in an attempt to yield the same results. Reproducibility really should be at a minimum, condition for any finding to be believable and informative. So with that in mind, as I'm sure you may have noticed by now, the amount of published cancer research out there estimated to be irreproducible is between 50 and 90%. This basically means if you were to try and reproduce two separate studies from one of your colleagues, the odds are better you would not be able to reproduce either study than reproduce both. And that is a real issue. Even studies published in high-profile journals have been proven to be challenging or outright impossible to reproduce successfully. And this, of course, has massive downstream effects on other studies, both basic and clinical, using irreproducible key findings to inform future research paths. Now, this leads to massive wastes in time, resources, and funding, which we all know is incredibly difficult to obtain. It is estimated scientists in the United States spend around $28 billion each year on basic biomedical research that cannot be repeated successfully. So the real question is, what can be done? So reliability is key to making significant advances and improvements in medicine. And right now, this is something that is lacking on a global scale. So what can we do to ensure the ongoing reliability of our own work is able to be reproduced? And the answer itself is difficult, as there is no single solution that can have this effect. Instead, we need to be focusing on a combination of experimental design improvements and procedural developments when undertaking our laboratory processes. So here are some of the ideas that won't actually cost you anything, but I believe are some of the most important things to consider if you really do want to curb the reproducibility issue in your own lab. First, don't read between the lines. As researchers, we all want to make groundbreaking discoveries that will change the medical world forever. And we also want to ensure we put our funding to good use. Ultimately, we're determined to see a result, and the notion of observer bias suggests that we may try and force this result even subconsciously. For an experiment to be reproducible, we need to observe only what is in front of us rather than trying to interpret the results based on our own preconceived ideas. Studies show that there is a definite difference in result interpretation between blinded and non-blinded research, which emphasizes the idea that many of us see what we want to see rather than what's actually there. So number 2, be methodical and be strict. Procedural flaws are one of the primary reasons why studies are frequently irreproducible. This suggests that a more rigorous approach to experimental design through hypothesis testing research, for example, is urgently needed. We need to be sure that the experimental design itself can be replicated using the information available. Just consider this. A review of a particular animal-based study found that only 9% of reproducers were able to replicate the experiment accurately. And this was due to the vagueness surrounding the experimental design. This, in turn, created a high level of uncertainty that ultimately led to invalid analysis. So try and be strict, adhere to guidelines, and don't deviate from the plan. Number 3, keep things transparent. Data disclosure is a massive problem at the moment, even despite the introduction of the full data disposition in recent years. So another example, there was a study that aimed to replicate the results from 18 gene expression experiments. The replicators here only managed to successfully reproduce 2, claiming the lack of available data was primarily responsible for irreproducibility. And just as an aside, this wasn't a one-off event. When 500 papers were reviewed, it was found that 59% of those published in journals that required full data disclosure did not adhere to the rules. The big takeaway here is if you choose not to disclose everything you are doing and the data produced, then you are absolutely at risk of having others not being able to replicate your research. It's as simple as that. And last, we have collaboration, input, and advice. And this cannot be stated enough, but don't be afraid to collaborate or seek advice from others when undertaking novel experiments. The more we talk and learn from others, the better the chance we have of completing the science we do correctly, efficiently, and ultimately making our own results reproducible. Beyond that, if you are unable to reproduce some portion of somebody else's research, it's always a good idea to reach out to see if there's something you may be missing, rather than just assuming that you or them did something incorrect. Okay, let's move on to areas that are a bit more specific that you as cell researchers can think about to make your research more reproducible day to day. So the first item on the docket is understanding microenvironments and how they can affect cells. And a generic definition of a microenvironment is something like the local environment surrounding a cell, which contains the physical and chemical signals that can influence cellular behavior either directly or indirectly. And this can include many variables. We're talking about things like media or number of cells. But for the talk today, we are almost going to exclusively focus on the chemical or gaseous microenvironments we subject our cells to. In this section, we have 3 major areas I want to cover. First, I want to look at CO2 incubators as a whole. And here I want to discuss how we grow cells within these systems and some of the challenges faced from a reproducibility standpoint. And then I want to try and look at some simple solutions that may help solve some of the issues that we discuss. Moving on, I will focus on one of the largest issues with working in CO2 incubators, which is not controlling for physiologically relevant oxygen levels. This is a huge factor within all cellular research systems, but especially so for cancer research, and I will discuss this, of course, in more detail. The last section we will touch on is what microenvironment information should be reported in publications to make the research you do fully reproducible. So regardless of what procedures you are doing, others can follow your methods and get the same results. Okay, let's get started. So what do we know about the air we breathe? Well, it's primarily made up of nitrogen at 78%, oxygen at 21%, and then we have trace amounts of all the other gases. Now let's quickly look at how we've been growing cells in standard CO2 incubators. Well, we culture them at 37 degrees Celsius to keep cells at physiologically relevant temperatures. We grow them at 5% CO2 to maintain pH, and at high humidity levels to slow evaporation of costly media. And to be fair, this has been working excellently for the last 70 to 80 years, and we've been able to culture almost any cell types in systems like these. We at Syntica even offer a standard cell incubator from RWD that can be used for this purpose. Just quickly, this system has been designed with simplicity and user functionality in mind and does an excellent job at growing and maintaining cells. This incubator, much like the ones you probably have in your own lab, does have one common issue that many researchers often overlook, and that is when the door is open, cells are exposed to room air, which is considerably different than the incubator environment we just talked about. Okay, so here's a graph within a standard incubator showing what happens to CO2 and temperature every time a door is opened. And it's pretty clear to see that both levels are changed drastically, with temperature dropping about 12 degrees and CO2 dropping its entire 5%. Now, if this was an infrequent event, it may not be noteworthy, but consider how many times a day in your lab different people are in and out of this oftentimes shared space. Every time the door is open, the cells will almost certainly feel some change in temperature and may even see some loss in CO2. Of course, this all leads to cell stress. So how can we combat what we see on the face is an unfixable problem? Well, first, if researchers in a lab are cognizant of this problem, they can actually attempt to open the door fewer times and try to actually limit the amount of time the door is ultimately opened. As we can see, incubators can get back up to set points pretty quick and can get cells back to the limits that they grow in best. Further, researchers can attempt to work on their cells diligently while outside of the incubator as well, again to limit cell stress in atmospheric air. But as you may notice, this doesn't actually solve the problem with the opening of the incubator door. To help, Synteca does have two options, which many researchers use to both open the door less and even remove atmospheric air from getting to cells. So first, we have the Condo Cell unit, which when placed inside of an incubator will circulate that air into the condos, which are the large white rectangles you see here. Each condo can fit a few petri dishes or multi-well plates. And when the incubator door is open, the circulation of incubator air into these units is halted, thus keeping the cells at the same condition over time. Further, condos may be removed from incubators and are able to keep set gas and temperature ranges for up to 20 minutes. Now, because the tops and the bottoms of the condos are clear glass, you could do most kinds of imaging on your cells and then get them back into physiologically relevant conditions in due time. Okay, so I'm just going to kind of quickly show you this video here. Just going to run you through this super quick. And it's a video of the condo cell so you can better appreciate the size and just kind of how this works. Perfect. So here's the general size of these. Again, the tops are glass, and that conditioning unit within is what controls the flow of incubator air into these units. Again, they are quite small and portable solutions, which are perfect for moving around to different microscopes or whatever you need to do. The tops and the bottoms are made of glass, again, for high resolution imaging. And last, they are quite small, which makes them very efficient for placing within any incubator. That's pretty much all I want to talk about with those. Perfect. Okay, so another option to limit the opening an incubator to check on cells over and over again is having a live cell imaging scope directly within the incubator itself. Here, Synteca offers the Lux 2, which is our live cell imaging microscope. And the idea here is a researcher can keep an eye on an assay or confluence over time without the need to check cells manually every day or every few hours, depending on the assay. This should help greatly with needing to open the incubator door on a frequent basis to check growth dynamics. Now, I will talk a bit more about the assays that can be done when I speak of automation, but keep in mind that cells can be examined from anywhere that has an online connection. In the moment we are in today, with COVID limiting the amount of in-person time and research is allowed within a lab, having the ability to document cell growth and movement from home or any location really is a huge positive to keep in mind. Okay, so the next part of this talk, I want to focus on what I think is the most under-considered microenvironment, not only in vitro cancer research, but in cellular research in general. So what do we know about oxygen conditions within standard cell incubators? First, and I only mention this because I've seen this mistake in several publications, is CO2 incubators are not at 21% oxygen. So what are they at? Well, they are much closer to 18%, as partial pressure of CO2 is at 5% and humidity is generally set between 70 and 90%, which means there's actually less room for oxygen within these systems. So let's keep that 18% oxygen level within CO2 incubators in mind as we look at a few relevant definitions next. So there really are four terms we need to keep in mind when we talk about oxygen in a cellular context. First, we need to understand that when I refer to normoxia, what I'm referring to is the air we breathe, which is at about 21%. What I need you to understand for this talk is when we refer to cellular oxygen, we want to get away from a human-centric way of talking about things and move to a more cell-centric point of view. And what do I mean by this? So this is an example of exactly what I'm referring to as cell-centric in terms of oxygen microenvironments. As you can see, while the air we breathe is at 21% oxygen, the air in the trachea is already down to about 19.7%. Once we're in the alveoli, we're down now to 14.5%, which is already more than 3% away from the 18% oxygen of a standard CO2 incubator. And as we continue to look at various tissue types, it only gets further and further away from this 18% oxygen. So back to our definitions. Hypoxia in a cell-centric approach means oxygen levels are below the standard growing conditions dependent on cell type. Alternatively, hyperoxia means oxygen conditions are above the standard microenvironment conditions, and Physoxia really refers to growing cells at oxygen levels that would be expected in the in vivo condition. So why bother with oxygen levels at all in vivo, or in vitro, sorry? Well, first and foremost, several models and research fields actually lend themselves directly to understanding how oxygen affects specific cell types. This includes most cell cancer models, sleep apnea models, stroke models, reactive oxygen species, and so on. Now, while those models lend themselves directly to oxygen playing a role, I also think oxygen levels are important to almost all cell types. So again, we can see here that while the air we breathe is at 160 millimeters of mercury, as we look at various cell types, they naturally grow at much lower levels. At the bottom here, I have included the fact that most tumorous tissue grows between 1 and 4% or from 7 to 28 millimeters of mercury. So what are the implications of growing cell types at high oxygen levels like 18% or 141 millimeters of mercury in a standard CO2 incubator? So hopefully I got this point across earlier, but I'll say it again. Normoxia does not equal Physoxia, and growing cells at higher oxygen levels is absolutely considered hyperoxic to cells. So the real question to consider then is, how do cells see hypoxia, Physoxia, and hyperoxia? And for this, we use the cell's natural oxygen sensor, the hypoxia inducible factor, or HIF for short. So super briefly, what we can see here is two separate scenarios. The one on the left basically shows that in a high oxygen environment, HIF becomes labeled for and is degraded in the cytoplasm. Now, if we look at the diagram on the right, in a lower oxygen environment, HIF in this case is not degraded and is inevitably brought back into the nucleus as an activator of transcription. So again, the question you may be asking is, so what? Well, here are a few of the many cellular systems that HIF drives the gene expression within. And this includes things like matrix and barrier function, inflammation, increased oxygen delivery, angiogenesis, anaerobic metabolism, cell proliferation, and so on and so forth. So basically what I'm suggesting is if this transcription factor itself affects a plethora of systems within a cell, and if you are not accounting for it, there's a good chance you could be missing something or adding a completely unnecessary variable into your study. Also, since your cell is closer to its natural state, you are now in a much better position to assess what it may actually be happening in the in vivo state. Here, I've just chosen a paper by Jing et al. 2019 to make a couple of points before I close out this section. So the image we are looking at here is a diagram demonstrating some of the systems that cellular oxygen affects that may lead to drug resistance in many cancer drugs tested. This includes systems affected by HIF, like apoptosis and autophagy, as well as some ancillary effects around lowering of pH, like ion trapping and genetic alterations. With this in mind, I think there really are two important points to take from this. The first point I want to make is something that I hope is understood by now, as I've stated a few times. And that's that growing cells far outside of physiologically relevant conditions adds a variable into any study that is often difficult to square, as HIF affects so many cellular systems. And when we speak of cancerous tissues that grow even further outside of this normal oxygen range, there is even a higher potential to miss the response that we are after. Point two, and just as important, is as a researcher, we want our cellular data we obtain to be a good proxy for what will be seen in future animal and hopefully clinical studies. If a factor as large to cancer as oxygen is missed, then you are adding a variable that will be difficult, if not impossible, to account for. So really, it is certainly something you want to think about as you plan your future studies. Okay, this moves us perfectly into the three options that Syntica has available for physiologically relevant cellular research. The three products we're going to touch on here are the Oxygenie, Foxbox, and HypoxyLab. And beyond that, we will delve a bit further into the HypoxyLab and its add-ons, just to give you a better idea of the functionality available to any researcher. Okay, so the first option available is the Oxygenie. And the Oxygenie brings researchers a small, portable, and continuous oxygen-controlled environment for short-term physiologically oxygen and temperature-based studies. Now, it's the ideal system for anyone needing to conduct high-resolution microscopy or irradiation under physiologically relevant conditions. For cancer researchers needing to irradiate their cells, it really is as easy as moving the small cup that you see in the bottom image, filled with your cells and media, irradiating it, and then placing it back into the system. The 6 culture wells, which are on the top left of the device, fully facilitate completely enclosed physiological growth conditions. And these are seated on microscope glass, allowing for adaptation into nearly any experimental procedure. With a system like this, you are able to get results equivalent to research carried out within larger hypoxic incubators and workstations. And if you can get by with the 6 samples at a time, it really is a fantastic option. Okay, next we have the Foxbox, which is a dynamic oxygen and CO2 controlled system. Now, the system can be used on a lab bench paralleling ambient temperature or humidity, but alternatively, and the way it's most commonly used, is within a standard CO2 incubator mirroring those conditions. It is a perfect system for short-term hypoxia experiments where media does not need to be frequently changed. It is great for those looking to get Physoxia hypoxia without having to purchase a larger, more costly dynamic hypoxia chamber, instead just converting some of their functional CO2 incubator space. It comes in 2 components, which are the gas mixer, which would generally stay outside of the incubator, and the box portion, which would go within the incubator. Just here's another schematic of this, and it really is important to note 2 things. 1, the gas mixer can control up to 3 boxes at once, with independent oxygen and CO2 control within each box. Second, the user has the option to purchase a small or large box to fit their individual needs. Last, Syntica offers the option that I believe is the most dynamic and advanced system on the market today, which is the HypoxyLab workstation. The HypoxyLab produces accurate, reproducible, and physiologically relevant cell environments, both for short and long-term hypoxic studies. It is the only system that I am aware of that measures oxygen using partial pressures, meaning it is not only more accurate day to day, but makes it much easier to compare measurements taken in vivo, which more often than not are taken in partial pressures. Further, it is not a huge system and fits on nearly any benchtop, and this size also means it does not use a copious amount of gas. It has a HEPA filter, which scrubs the air completely within 30 seconds of startup as well. Now, of course, I am a bit biased as this is a system that we offer, but again, I do believe it is the most well thought out and dynamic system on the market today. So something to keep in mind, the system does have several add-ons which can be helpful to any researcher. First, we have the CytoSmart Lux 2, which we already talked about, and this can be placed inside of the system, allowing for the user to conduct various assays within. Again, we already talked about this as well, but the Condo Cell can also be used here. And this is really for users who want to use more advanced microscopy. The Condo Cell really allows researchers to take their cells out of the workstation, keeping them at the HypoxyLab set conditions for upwards of 20 minutes to do their imaging. It is an inexpensive option for researchers to image cells under hypoxia and is oftentimes bundled with the system. The last option we have is the OxyLite system you can see here. The OxyLite oxygen monitoring system will provide a method to generate highly accurate measurements of dissolved oxygen directly from within cell cultures and media within the HypoxyLab. Now, it does this via a miniature fiber optic sensor that is passed into the chamber through a dedicated side gland. Measurements with the system are as easy as placing the sensor into media for a couple of seconds, And we will see this actually in the upcoming video. The sensor itself does not consume oxygen, which means it will be more accurate over time. Last, the OxyLite can be used in vivo as well for live tissue oxygen measurements. So again, here's just a quick video of the HypoxyLab. Just going to jump about 10 seconds in here. Perfect. So again, here's the HypoxyLab I just spoke about. Again, it has a pretty small footprint. Any user can work within the workstation itself, which makes it very, very easy to keep your cells under hypoxic conditions. The setup of the system is really easy. I've done this a few times in under 30 minutes. Again, one of the biggest things about the system is it uses partial pressures as opposed to percentages. The digital touchscreen is extremely easy to use on this system. It has internal lighting, which again makes seeing everything easy. And this is also really nice. It has a rapid transfer hatch, which allows you to get your samples in and out quite quickly. Again, HEPA filtration is standard on this system. And again, we're going to now see a couple of the extra things. So data logging, and then we'll move on to look at yes, there's the OxyLite measurement. And again, we're going to actually see the probe itself being placed into the media. And again, it would take an immediate measurement. Also, the CytoSmart can be used within this system. Perfect. Okay. Now let's just move on to the last section of microenvironments, which is how and what to report so your work can be reproduced. And basically here, I'm summarizing one of my favorite papers on this subject. And the paper is called Oxygenation in Cell Culture. Critical parameters for reproducibility are routinely not reported. And this was published in 2018 by a group out of the University of Calgary. I cannot highly recommend enough that anyone doing cellular studies read this paper and follow what is laid out here, as it will undoubtedly increase the chance that anyone else following your research can reproduce it. So the group here focuses on the items that I've listed here in regards to understanding microenvironments in cells and making research reproducible. So first, the first thing to look at is temperature in which cells are kept at. This is important as different temperatures change how fast gases can diffuse into media. Next, researchers should try and get in the habit of including the atmospheric partial pressure of where they are. So this is almost always overlooked, but different altitudes change the equilibrium of dissolved oxygen. So the group in this paper actually uses the example that they are in Calgary, which is situated at 1,045 meters. And the atmospheric pressure is about 13% lower than sea level. Now, if this was not taken into account by other labs at lower altitudes, the amount of oxygen that cells see as a percentage would be considerably off from what this group actually shows. Now, there is a slight caveat to this, and this is if you use a hypoxia chamber that takes measurements in partial pressures like the HypoxyLab, you'll never ever have this issue. But it is still good practice, and I do recommend including local atmospheric partial pressures. Moving forward, the tissue culture and media composition can also affect diffusion of gases. And here, increases in the ionic strength of culture media can reduce the solubility of oxygen and speed at which gases will diffuse. As we talked about, handling and removal of tissue culture is also important. As we know, opening incubator doors can change the gas mixture within an incubator. And within a CO2 incubator, this can take a few minutes to adjust, and within an oxygen-based system, it can take up to an hour. Now, this causes changes in oxygen concentration within the cell culture media, and this is particularly noticed in experiments in low oxygen atmospheres. Next, culture vessels should also be reported, as different vessels allow for different amounts of diffusion. Specifically, which vessels are used, and keep in mind that the shallower the media and the more surface area available to the environment, the faster the diffusion of gas will be into the media. And last, the last three of cell type, seeding density, and media height all play a significant role in the amount of diffusion and the time needed to reach specific gas levels within the media. So while this may all sound pretty intuitive, the group here also looked at some of the most recent cellular publications in top journals and found quite definitively that most publications were missing two or more of these factors above that would be needed to make the research fully reproducible. Now, on a side note, we are slowly beginning to see a bigger push by these journals to do a better job of making sure their method sections of in their papers contain more of this type of necessary information as replication issues are brought to the forefront. Okay, so just to give me a tiny bit of a break here, we are going to ask another audience poll. And the two things we would like to learn here from you is, do you think the constant opening and closing of incubator doors could be a source of irreproducibility in your lab? And the answers here are: we are diligent and we take caution. We perhaps need to look at this a bit more as a source of irreproducibility, or it's likely an issue and we do need to address this. And secondly, are you currently using physiologically relevant oxygen conditions with your cells? Yes, we are. No, but perhaps we should look into this, or no, it's not relevant. And we're going to take about a minute on this, and I'm just going to have a quick drink of water and we'll start the second half here soon. Okay, guys, it looks like mostly everybody's done here. I'll give it another few seconds. Okay, wonderful. Okay, so we are now moving into the latter half of our presentation, which will focus on automation and its effects towards solving the reproducibility issue. So reducing human error is perhaps one of the best ways to improve reproducibility, and science is becoming more and more automated. In recent years, there has been a huge number of startup enterprises in the United States promising to forever change the way data is collected through robotic processes. While many researchers won't be quite at a stage where they can fully automate their research, that doesn't mean we can't take some measures to increase automation within our own labs. Some of the most beneficial offerings seen today include automated microplate movers, automated incubators, as well as a wide range of softwares that help to automate data collection and organization for publication. And of course, we'll speak a few of those here. So here, we will briefly talk about why labs should automate procedures, and then we will focus on two areas of automation to help cancer research in particular. Now, this includes how to automate cellular assays through the help of live cell imaging, and then automating cell and colony counting. So why automate lab procedures then? Well, let's start with the simple fact that a well-designed automated system almost always improves the speed and accuracy of any task over a manual human performing the same objective. A machine can completely remove some of the human fallacies like bias or subjectivity, even if it's unwittingly. An automated process does the same task time and time again and should not suffer from a great deal of variation in whatever tasks it undertakes, whereas humans can often suffer from fatigue or intra-observer variation. By removing the human variable, however small or large it may be, the overall science being done should be considerably more reproducible if the same machine variables are used across an entire study. So let's look at this in practice with a system we saw earlier in the LUX2 and its dynamic sister system, the Omni, for enabling automation of cellular assays. Okay, so on the right we see the LUX2 on the top, and below it the Omni microscope. As discussed, these can be placed directly into an incubator or workstation to facilitate looking at cell growth dynamics in more physiologically relevant conditions, thus removing atmospheric cell stress from the equation. With these systems, time-lapse movies are automatically generated and can be assessed immediately. Now, you can obtain real-time updates of your cells even during image acquisition. These images, as mentioned earlier, can be assessed remotely anywhere that you have an internet connection, which will free up lab space for others. This also allows researchers to manipulate cells when absolutely necessary for things like cell splitting. Further, notifications can even be set up and sent out to lab members when confluence reaches specific percentages, again, helping to make research more efficient when subculturing. So since you may be wondering, what are the major differences between the Omni and LUX2? Well, the most substantial difference between the Lux and the Omni is the Lux has one focal point, whereas the Omni can image an entire plate in exquisite detail, and I'll demonstrate both of these in a bit. Before that, the last point I have is these systems can be used to automate various assays, including wound healing or scratch assays, confluence measurements, morphology studies, cytotoxicity assays, proliferation assays, single cell migration, and even more. So there is quite an array there. Next, I just want to quickly run through 3 separate short videos, with the first 2 being for the Lux and the last one demonstrating the Omni microscope. Okay, so what we're looking at here is a time-lapse video of proliferating normal human epidermal cells from an adult donor. And these images were taken at 5-minute intervals across 73 hours. Now, this video looks very fluid to me, but again, depending on your internet connection, it may be a little less fluid, but you should be able to get the idea of what we're doing here. That's probably enough of that. Okay. And the next video I have is a 36-hour time-lapse video of a scratch wound closure by 3T3 cells. And these images were taken at 15-minute intervals. So as you can see, the wound is closed, and again, the microscope continues to image. Okay, so the last one here is a bit more of a promotional video for the Omni, but really demonstrates the power of this type of system. So the first thing, and I'm just going to jump forward a little bit. So the first thing you're going to notice is the Lux is able to image a much larger area, in this case, a 6-well plate. And the software itself is able to make a multitude of measurements. So here, again, we can see confluence across each well. Then what we can actually see is we'll do a bit of zooming in here, and it's going to actually zoom again to give us cell dynamics. So here's a piece of debris, and we'll move forward, cell clustering. And moving forward again, we can see cell morphology, attachment, and cell division. Now, what's really, really neat is we can look at one area of interest, and we can actually speed through time, again, getting videos. Of course, we can come out at the end and look at our confluence over time, if that was your primary objective. So that's just to give you a pretty good idea of what that system can actually do. Okay. So moving on to the last section of this talk, I really want to focus on another area cancer researchers in particular can automate to save time and to get more consistent results, and that is with cell and colony counting. Now, both cell and colony counting have similar drawbacks when done manually. They are slow and painstaking tasks to accomplish, and further, they can be error-prone and suffer from variability from count to count. Now, I will touch on these points ahead, but counting cells or colonies with the use of machines actually removes bias, variability, subjectivity, human error, and has been proven time and again to save time, effort, and cost for labs in the long run. Okay, so cell counting reproducibility. So for decades, biologists have relied on counting cells using a microscope slide called a hemocytometer, which you can see here. The slide holds fluids at a fixed volume, and cells can be counted using a microscope. At present, automated cell counters are becoming more popular for their ease of use and the rapidness of results. So why should you consider automating this process? So first is human perception of what defines a cell. Manual definitions and recognition of a cell versus cell debris or other particles can be challenging even for the trained eye. Each person performing the manual cell count adheres to a certain set of criteria that define what is a cell with stain and what is not, especially in viability studies. Such variation in human perception when counting manually can be extremely detrimental to experimental setup and analysis when counting cells manually. If multiple users count the same sample, it is not uncommon to see a significantly high variance in counts. Next is volume dilution and pipetting errors. And the preparation and loading of the cell samples in a hemocytometer can actually give rise to errors. Though the hemocytometer itself contains a given volume, the space between the counting chamber and the cover glass can be slightly increased or decreased when filling the chamber with liquid. This can cause errors based on estimating volume incorrectly, and volumetric inaccuracies can also arise from pipetting or from serial dilutions. So next is viability determination, and what we're talking about here is looking when using trypan blue. So when manually estimating cell viability, trypan blue is used as a marker for dead cells. Since the intensity of the stain can vary, it can be quite difficult to determine whether a cell stains positive with trypan blue or does not in a given sample. Now, given that trypan blue is toxic to cells, viable cells are eventually stained if not analyzed in a certain time frame, depending on sample conditions. So for these reasons, trypan blue is known to underestimate the viability of cell populations, and caution really should be used when trying to interpret trypan blue-based measurements in cell viability manually. Now, why should researchers perhaps consider an automated cell counter like the one seen on the right, which is another item Synteca offers from RWD? Well, first, automated systems have no human bias to influence results. The software algorithm defines a cell based on the same set of variables, and it does this constantly for every analysis. Next, there is no human errors in calculations, as the cassettes supplied have a pre-calibrated volume for precise and reproducible results. Next, these systems offer significant data generation. And beyond this, the instruments themselves will even alarm you if you have too few or too many cells in a sample, and this further reduces the chance of error. Last, the time savings really are significant, as a researcher can place a cassette into an automated cell counter and have that sample analyzed and counted in under 20 seconds, which is easily minutes ahead of the fastest hemocytometer counts. So with that, let's move on to the last area I wanted to cover today, which is colony counting. So the clonogenic or spheroid or colony counting assay, as it's commonly known, often requires that a single well-trained individual counts hundreds or thousands of plates to reduce the chance of variability between counts. Like cell counting, it falls victim to many of the same issues with interpretation, like what is the lower threshold of a colony for it to be called a colony? Further, if counting 3D colonies under a microscope, there's also the issue of needing to count across the X, Y, and Z axis and try to avoid missing or recounting colonies along the way. Again, it's a highly painstaking process that lacks any standardization and does not really provide any additional information about the colonies, like size information, which oftentimes is even apparent to those counting between different samples. Further, while the colony-forming cell assay is known as the gold standard method for measuring things like radiation, chemotherapeutic drugs, and other agents on cell viability, it is also known to be a costly time investment for any lab undertaking this assay. So why automate these assays? Well, automating the detection, counting, and analysis of mammalian cell colonies offers significant benefits to cancer biologists processing tumor colony forming assays, which eliminates the risk of subjectivity, bias, and human error, while increasing the speed and accuracy, all while delivering unprecedented data archiving and retrieval capabilities. With its dynamic effect on throughput, automated colony counting opens up the possibility for ambitious new products that will advance the frontiers of cancer research. And this includes large-scale combinational studies. Furthermore, today's intelligence-based systems for automated colony counting enable the collection, processing, and archiving of digital samples, again, adding a powerful new dimension to the peer review and reporting process. They also deliver additional quantitative data and make innovative collaborative research and validation approaches possible. Okay, so here's the system that we offer for automated colony counting, and it is the GelCount by Oxford Optronics. And it comes in two parts, and as you can see on the left, there is the hardware component, which images, and the software component, which does the analysis. And what I'd quickly like to do is just show you how fast and efficiently we can count one 6-well plate of 3D colonies. So here I'm actually just going to quickly move to share a different screen for you. And this is actually the software itself. And I've actually done a previous webinar on this, which I go into extensive detail, but I just wanted to quickly run through how quickly I can do this. So first I can actually load a mask. And all a mask is the green circle here. And basically the way this mask works is anything within the mask gets counted, anything on the outside does not. And I've preset this mask on every well of this 6-well plate. Next, we want to be able to actually count the colonies within. And for this, we actually use this optimizer function. And you can see there's several tabs here in which we move around the different settings to count the colonies. But again, once you've previously done this, you can actually load your previous settings. So I'm just going to bring up those quickly. We can load our previous settings, and we can actually count this one well. So that'll take a couple seconds. So you'll notice we've only counted one well of the 6-well plate. And the last step, we can actually recount all. And what you'll notice is each of these wells will turn red as it counts each one. So there's 1, 2, 3, 4, and 5. So perfect. Now every well has been counted. The other thing you'll notice is that all of the information that you might need, like counts, diameters, areas, densities, volume, nearest neighbor distance, are all located on the side. But I will show you that you can also save these to an Excel file coming up. Beyond that, that just shows you just the relative quickness of a system like this over manually counting thousands of colonies. Okay, so back to the PowerPoint. Okay, hopefully you guys are back to the PowerPoint, and let's move forward. So here's the type of data that we can get within an Excel sheet. And here we are simply looking at the overall well-to-well data that we saw before. And so we're talking about things like total counts, diameter information, and nearest neighbor distance, along with their respective standard deviations. Also important to note is information like the saved mask and algorithm settings that I just showed you are also provided, so there's no guessing weeks or months down the road about which setting you used in a particular experiment and which to use in your follow-up experiments. Now, what I think is even more important and useful to researchers is you also have access to the full data from each and every counted colony in a respective well or dish. So here is well 1 and well 2, for example, and just so you can follow along, each line on this Excel sheet is the individual information of one counted colony. Now, what this means to you as a researcher is you actually have the ability to run multivariable statistics, across treatments comparing things like survival, volume of colonies, and nearest neighbor distance, which just is something that wasn't possible with the manual counting of colonies. And this can take what would normally be a simple colony viability study to a much more dynamic experiment by looking at a range of factors and their effects. So the last thing I really want to show you about this system quickly is I just want to show you that we can get very nice images of our plates processed and unprocessed, which is helpful for putting together things like presentations, posters, and publications. It also demonstrates to others in your field that you are appropriately counting your colonies and can act as a nice check and balance to your research. It also makes your research look more reproducible. So just to summarize, we talked about many things today, but first we looked at the reproducibility problem and its massive cost to research in general. We then briefly discussed some simple solutions that any lab can take to increase reproducibility. Next, we focused the discussion to microenvironments, and we looked at ways to lessen shocking cells within an incubator, and then discussed the importance of physiologically relevant oxygen conditions in cancer research. Last, we discussed automation and how beyond making research more reproducible, it can also offer a plethora of other advantages to researchers. Finally, I do have a couple parting thoughts for you. The ultimate aim of improving reproducibility is not only to enable better and easier replication of our own work, but also to allow others to duplicate and confirm our results and findings. Reproducibility is essential for all areas of research, but it is understood to be especially important for basic and preclinical studies, which, as we know, often form the basis of further research into specific areas of interest. Unfortunately, even if we do take measures to improve the reproducibility of our own work, the issue still remains that there are very few set guidelines regarding reproducibility. Even the guidelines that are currently present appear to be rarely enforced, such as journal requirements for data disclosure, which seem to be at the discretion of the publisher. As we have now entered a period which many are calling the reproducibility crisis, I hope that this will force these changes in this industry within the next few years. How these changes will occur is unknown, but many are expecting funding bodies to be called into question, potentially asking for these bodies to include reproducibility as a requirement and incorporate it into publication criteria in the future. So with that, I wanted to thank you for watching this presentation. I hope you found it educational, and I hope it's allowed you to think a bit more about your future research goals. I am going to quickly cover a few of the questions that I can see are coming in, and please feel free to ask now if you'd like as well. And I'm going to have a quick drink of water, and then I'm just going to start those questions. Okay. So one of the top questions I have here is, can the Lux microscope that you showed be used in any hypoxia system? So the answer here is maybe. It will most definitely usually work in most standard CO2 and tri-gas incubators, and there should never be a problem there because they have dedicated ports. But other workstations outside of the HypoxyLab would have to be looked into in an individual basis. We really just need to make sure we can get the power supply within. So that would be the first thing. How long would you suggest degassing media to get to a specific set point? So again, this question is highly variable and dependent on your needs, but I do have a few suggestions to degassing media more efficiently. So I mentioned this in this talk, but surface area definitely matters. The more contact the liquid you are using has to the, let's just say, low oxygen environment, the better. So I often suggest using a T25 flask for degassing versus beakers, or even worse, an Erlenmeyer flask. Similarly, the depth of media also plays a role in degassing, and again, shallower here is better for degassing. So again, T25 flasks work great. The last point I do want to make is, if you do need to know the levels that your degas media is at, again, a system like the OxyLite where you can place a sensor within can help you figure that out very, very quickly. Can the colony counter count stem cell colonies? This actually kind of depends, and it's more on a case-by-case issue, but the GelCount will not dependently detect highly diffuse colonies or highly irregular colonies. The algorithm itself was not made for colony counting based on morphology. So I would generally want to see what you're working with, and I'll probably reach out afterwards and just ask you what you're working with, and we can make a determination pretty quickly. Let's just go through a few more here. What about 3D culturing? Is one of them suitable for 3D culture imaging? Yes. So this question here, so about the 3D culturing, that should be, the system that should be most appropriate for that would be the GelCount that we talked about, for counting them at least. And then for growing your 3D cultures, again, I always suggest doing it within an oxygen environment that's relevant to what you're doing. So I think that's probably the best answer for that. Just looking at the time here, I'm just going to probably take one or two more questions here. Our lab uses Petri dishes for our assays. Can colonies be counted using Petri dishes? So the answer here is yes. As far as plates, the GelCount can do 6 to 96 well plates. It can also do 35, 50, and 100 millimeter Petri dishes, and some T25 flasks as well. And let's just make this the last question for the day so everybody can get on their way. Does the Condo work in tri-gas incubators or just CO2? And again, I think I mentioned this, but the fan system in the Condo Cell is set to push whatever environmental conditions that are around it into the condo. So again, when the door is open, whether it be a CO2 or tri-gas incubator, the condos will be shut off for a defined period of time. So, and again, then they'll start up again probably 20 minutes later to push the atmosphere in again. With that, I think that's probably where I want to end today. We have pretty much reached the 60-minute mark of our session, so just to be respectful of everybody's time, I am going to wrap things up. As mentioned at the start of our webinar, I will be sure to answer any outstanding questions you guys send along privately. And then I'll also send out that Q&A document in the coming days. Do feel free to reach out to us directly if you have any questions about anything presented today or any of the items that we talked about. And last, I just want to thank you all again for taking time out of your day to come be with me. And I'm very happy you attended our session, and we hope to see you in future sessions. Thank you and have a nice day.